The cerebellum is functionally distinct from the midbrain and hindbrain regions, as well as from the rest of cortex. For a long time, the cerebellum was thought to be exclusively involved in motor activity, since cerebellar damage leads to significant impairments in motor control and posture. The cerebellum does play a key role in motor programming, although it doesn't generate motor activity itself. Rather, the cerebellum modifies both voluntary motor commands coming down from the cortex to the spinal cord and autonomic motor influences coming from the brainstem, fine-tuning each based on environmental context and learned outcomes. As such, the cerebellum is thought to be a major facilitator of motor learning. More recently, scientists have discovered a key role for the cerebellum in certain cognitive functions, like language. This role is not at all well understood, and as such, our discussion will focus on cerebellar circuitry that's been studied in the context of motor learning. But you should be aware that this is an ongoing area of research, and be on the lookout for updates on the cerebellum's broader role. Partly because we have such a limited understanding of its functional properties, an intensive amount of work has gone into characterizing the structure of the cerebellum. In fact, unpacking the cerebellum's morphology is considerably easier than comprehending its function, since the cerebellum has a highly unique, highly regular organization. Even a quick glance will tell you that the cerebellum is distinct from the rest of the brain. The cerebellum, Latin for little brain, appears as a separate structure attached to the bottom of the brain, looking something like the ridged head of a cauliflower. You shouldn't be fooled by its meager appearance, however. The cerebellum is a true neuronal powerhouse. While it accounts for only 10% of the brain's volume, the cerebellum contains at least 50% of the brain's total neurons. Interestingly, this ratio seems to be conserved across mammalian species. Cerebellar neurons are packed into many tightly folded layers of cortex, which wind in deep petal-like grooves around a white matter core, and converge on a fluid-filled ventricle at their base. These folds, though intricately patterned, are quite regular. They're reproduced across individuals and highly stereotyped in their organization. At the heart of the white matter lie 4 distinct clusters of cell bodies. These are the deep cerebellar nuclei, and they're the only part of the cerebellum that sends output to the rest of the brain. Although the cerebellar cortex's neuronal network is highly intricate, it's also highly patterned. 2 types of neurons appear to play particularly dominant roles in cerebellar circuitry. These are the Purkinje cells and the cerebellar granule cells, both highly specialized neuronal populations, whose distinctive morphology sets them definitively apart from the neurons found in the rest of the brain. Purkinje neurons are a class of GABAergic interneurons with very large and characteristic dendritic arbors. They are some of the largest neurons in the brain and function through 2 distinct forms of electrophysiological activity, which we'll investigate in a moment. In contrast, the granule cells in the cerebellum are the smallest neurons in the brain and send out only 4 or 5 dendrites per cell. These dendrites have characteristic enlargements at their ends known as dendritic claws and are the sites of excitatory and inhibitory input from other neurons. The strength of the granule cells is in their numbers, since they are by far the most numerous neuron in the cerebellum, hugely outnumbering the giant Purkinje cells. In fact, in humans, the average number of cerebellar granule cells is around 50 billion cells, which means they constitute about 3/4 of the total number of neurons in the brain. The granule cells are functionally distinct from Purkinje cells. They're glutamatergic and excitatory, unlike inhibitory Purkinje cells. The 2 cellular populations also occupy distinct regions of the cerebellar cortex, which is made up of 3 layers. The innermost layer contains the nuclei of the 50 billion cerebellar granule cells pressed in against the deeper white matter. Consider that this single cerebellar layer contains over half of the brain's cells. You begin to get an appreciation for just how tightly packed the cerebellar cortex is. The next layer contains the cell bodies of the Purkinje cells, which is just 1 cell layer thick. Finally, the outer layer of the cerebellar cortex contains the axons of the granule cells, as well as densely branching dendrites from the Purkinje cells. In addition to the cell bodies of the Purkinje and granule cells, the cerebellar cortex contains 3 prominent types of axonal projections. These are the mossy fibers, climbing fibers, and parallel fibers, and their interactions with the Purkinje and granule cells mediate key functional processes within the cerebellum. Mossy fibers and climbing fibers provide inputs to the cerebellum from the rest of the brain. The mossy fibers originate in the brainstem and make excitatory connections onto cell bodies in the deep cerebellar nuclei, as well as onto the granule cells in the cerebellar cortex. Each individual mossy fiber exerts widespread influence. Indeed, a single mossy fiber innervates hundreds of granule cells. The second type of axonal projection, the climbing fibers, come exclusively from a particular region in the medulla called the inferior olive. These fibers have long axons which climb up the dendrites of Purkinje cells like ivy. Each Purkinje cell receives one very powerful input from a single climbing fiber. Indeed, although each climbing fiber contacts only about 10 Purkinje cells, it contacts each of these cells in about 300 different places. Thus, the brainstem provides two types of input: widespread influence of many granule cells with each mossy fiber, and powerful control of a few Purkinje cells with the climbing fiber. Actually, both types of input have their end effect on Purkinje cells. This is due to the activity of a third type of axonal projection, actually that comes from inside the cerebellum itself. These parallel fibers are axons of the granule cells, which split into the topmost cortical layer, run in parallel to the folds of the cerebellar cortex, and perpendicular to the Purkinje cell dendritic arbors. This particular organization allows each parallel fiber to come into contact with hundreds of Purkinje cells as it runs through the cortex, and each Purkinje cell is contacted by thousands of parallel fibers. Since mossy fibers control broad populations of granule cell firing, they have a significant influence on pyramidal activity as well. In fact, while acting disynaptically through the granule cells may seem inefficient, it actually greatly increases the power of each mossy fiber, since parallel fibers carry the mossy fiber signal far and wide. This complex circuitry is highly specific to the cerebellar cortex. The only way that the information it encodes leaves this unique region is through the Purkinje cells, whose axons synapse onto the deep cerebellar nuclei, which in turn are the sole route through which signals leave the cerebellum as a whole. Since the Purkinje cells are GABAergic, they exert an inhibitory influence over the deep cerebellar nuclei. Due to consistent parallel fiber stimulation, the Purkinje cells reliably fire about 70 spikes per second, tonically inhibiting the cerebellar nuclei. Much rarer is the activation due to the climbing fibers, but when these activations occur, they're highly powerful and can induce long-lasting changes in the excitability of Purkinje cells. Indeed, climbing fiber-mediated changes in the strength of parallel fiber inputs to the Purkinje cells are thought to underlie motor learning in the cerebellum, making pathways corresponding to correct behavioral paths stronger than those corresponding to weak ones. Climbing fibers fire specifically when a desired motor outcome does not correlate with the actual motor behavior that an individual produces, to weaken the strength of paths corresponding to the wrong motor behavior. As such, the next time the same behavioral context occurs, the incorrect behavior is less likely to be repeated. Mossy fiber and climbing fiber inputs thus provide fiber-specific information to facilitate learning of particular motor programs. However, the cerebellum also receives global modulatory input through dopaminergic, serotonergic, and noradrenergic and cholinergic pathways. These diffuse neuromodulators have been shown capable of exerting a significant influence over cortical excitability and likely have a comparable function in the cerebellum. Thus, the extent to which the cerebellum is keyed to learn motor patterns may depend on the neuromodulation setting of a particular neural state. Given the cerebellum's demonstrated role in motor learning and modulation, it's not surprising that extensive damage to the cerebellum can have devastating effects on motor function. However, cerebellar injury has also recently been associated with impairments in language, attention, and mental imagery, and might be involved in the pathogenesis of several neuropsychiatric disorders, such as autism.